हेलो। हेलो। पार्थ भाई, मेरी आवाज आ रही है ना?
यस मैम, यस मैम।
ओके, थैंक यू। Good evening, Vasudha ma'am. We will shortly start as soon as the participant will join, and then we will unmute you. Thank you so much. All the participant who has now joined, good evening to one and all. You can chat once it is required on the raise hand option. We will unmute you. Thank you.
Thank you, mostly.
It's 5:49. I guess another few minutes and then we can start. Dr. Devashish sir did join. Devashish sir. पार्थ भाई मेरी आवाज आ रही है ना?
Yes, ma'am.
जी.
Good evening, madam.
Good evening, sir.
Okay. Can you please receive my call once?
No, sir. Because it is internet connected. If I do, my this connection will go.
Okay.
So can you please chat, sir? It is a humble request.
Yeah, not a problem. Just
Yeah. Thank you. Sorry for the inconvenience, sir.
Dear ma'am, kindly look at one in your chat, please, once. पार्थ सर, आप ऑनलाइन हो? हेलो।
Yes, sir. Yes, sir.
Okay, okay. Thank you, sir.
Okay.
Ma'am, are you there? Moshe, ma'am, are you there? Okay, so anyway, very good evening to all the
Good
participants.
evening, one and all.
Yeah, okay.
Am I audible to all of you?
Yes, yes, ma'am. Please continue.
Yeah. Good evening, dear participants, respected coordinator, Dr. Devashish sir, present here, backhand support team, and all my respected heads and authorities of Atmiya University present in this forum. Invoking the divine blessings of Lord Swaminarayan Brahmaswarup Sri Hari Prasad Swamiji Maharaj, Pragat Guru Swamiji Maharaj, and our beloved president and mentor of Atmiya University, Param Pujya Sri Tyag Vallabh Swamiji Maharaj. I welcome all of you in today's third day session of 6 days Atal FDP program. With this note, I would like to take a privilege to pray the God with 3 Om chant. Please close your eyes and with folded hands, all the participants are requested to do 3 Omkars. Let's start the session, initiating with introducing few important gentle reminders to all my beloved participants. Participants, you will be shared attendance link and feedback link in the chat box during the session. Kindly fill the attendance till the session ends today itself. Chat in the chat and also in the WhatsApp group, the feedback link will be shared. Till tomorrow, 12 noon, you will have time to share your important feedbacks, which can improvise our organization of such type of event in future. Second, you can chat your questions of the respective speaker related to the topic in the chat box. And if you wish to contact directly for the questions, we can unmute you with the raising hand options, which is available in your Cisco WebEx link. With these short things, I would like to take the privilege to announce today's speaker, Dr. Vasudha ma'am from Aristogene Biosciences, Bangalore. I would like to take the opportunity to introduce her vast experience of 25 years in a nutshell before you. I thought I can do my best. Madam has 25 years of experience in myriad domain of research. She is actually a biochemist. And apart from that, she has also done some areas of work on development of bacteriophage-based therapeutics for poultry, cloning and expression studies, enzyme modifications, product and process development, research education, kits development is also one of the area where she has excelled. She has guided a good number of PhD students, research students on various facets of researches like DNA fingerprinting, single nucleotide polymorphism detection, such types. With this brief overview of her vast credentials, I would like to invite Dr. Vasudha Madam from Aristogene Biosciences to take the stage online to share some insights on today's topic of her session entitled bacteriophage-based products, a promising solution for treating antimicrobial resistance or AMR in poultry, aquaculture, and livestock. Madam, all the stage is yours. Thank you so much for joining us and giving us the privilege for getting some insights from your side. पार्थ भाई, वसुधा मैम को प्रेजेंटर राइट्स दे दीजिए, उन्हें अनम्यूट कर दीजिए.
Good evening.
Good evening, ma'am.
And thank you one and all for this wonderful opportunity given to me. Okay. So today's topic already Mausmi ma'am has told you. I would like to share my screen. Okay. Are you seeing the screen?
Yes, ma'am. It's
Yeah.
Yeah, it's scaled, ma'am. Can you do the full slide, ma'am, please?
Yeah, it is full slide.
I mean, ma'am,
No, I think, ma'am, it is full slide only. We can see that. Okay, maybe because of your network, it has not come, but
okay,
we can see the full
thank you.
slide.
Yes, thank
Yeah.
you.
Ma'am, you can continue, please.
Okay. So today's my topic is what we are working from years, the bacteriophage-based products, which is a promising solution for treating AMR in poultry, aquaculture, and livestock. So at present, the global health threat, which is impacting humans, animals, and environment, is antimicrobial resistance, which is AMR. So let us define antimicrobials. They are the drugs which are used when we get infections, diseases, which is caused by bacteria, like microbes like bacteria, fungi, protozoans, etc. And we are most familiar with the very popular antimicrobial, which is antibiotic. And I'm sure all of us have used in our life, at least once in our life. You all agree with it. And so this popular antibiotic, since its discovery some 80 years ago, it has saved hundreds of millions of lives. So now these antimicrobials, why is it getting resistance? See, the pathogens which are continuously facing these antimicrobials or antibiotic in particular, they will learn how to overcome this problem, how to salvage the antibiotic itself with the changes, genetic changes in their genome. And it's a natural process. When an organism is continuously exposed to certain antimicrobial, it will learn how to overcome it. So that's how antimicrobial resistance has started. And to become so much a problem, where is it used and how much are we using? If you see this picture, 70% of the antibiotics manufactured are used in animals and 30% is used by humans. According to the estimate of the World Health Organization, only 50% of the antibiotics are used correctly. I'm talking only about the humans use. 30% of the antibiotic prescription was not required at all. And up to 60% of antibiotics in Africa and Asia may be substandard. And another problem, there are counterfeit drugs infesting the market, and also the availability over the counter. So whenever you have a small ailment, just go to the drug house and you can avail easily without any prescription. That's one more problem. So this overuse and abuse of antibiotic contributes to dangerous drug-resistant bacteria, which are called as superbugs. Superbugs meaning they will be resistant to many or all of the antibiotics used on them. And one more thing is that our country, India, is called as AMR capital, which is, it's not acceptable. Like people are calling India as capital of AMR. So now, how does this antibiotic resistance occur? So during infection, in the sense that the lot of bacteria are there and the antibiotics are given, during that course, what all happens? Initially, there are a lot of bacteria. A few of them learn how to overcome the antibiotic problem. They can degrade antibiotics. So in the course of time, the antibiotics acts on all other bacteria and they are killed. Only this resistant bacteria starts growing and now it has a very favorable condition. So it continuously and happily multiplies, and then further it can transfer its resistance gene to other neighboring bacteria also. So this is how antibiotic resistance occurs. And now how is it spreading? These farm animals, like in aquaculture, the shrimp, fish, poultry, pig, and dairy, they are all treated with antibiotics. It's a known fact now. Thoroughly, they are treated with antibiotics, and the antimicrobial resistance is majorly present there in the farms, and in two ways. One, directly in the meat, the antibiotic residues as well as the resistant bacteria can pass on and to the humans. And secondly, the animal waste, which contains again these residues and bacteria, will be added on to the used in agriculture. So from there also humans are getting the AMR organisms and also the antibiotic residues. So this is one story, the 70% story, and the 30% here, again, we are taking antibiotics knowingly, unknowingly, wantedly, unwantedly. If someone wants to go on a tour or some wound, antibiotics are taken. So the the resistance develops, and it again in the family it spreads. If it is increased, you cannot, you did not cure with your own antibiotic intake, you go to hospital, and in the hospital again the caretakers are getting affected. So in the hospital also it starts spreading, and we all know that ICU infections are so common there also. So whether you are cured or not cured, go back home, and this cycle continues. So this is how antibiotic resistance spreads. So what we need is at present the solution for these antibiotic resistant organisms. The consequences are like this. If you put together all the causative reasons for the deaths, including everything, like whether you will take it, diabetes, cancer, measles, even road traffic accidents. At present, the AMR is around 7 lakh deaths per year, and it is estimated that it can go up to 10 million deaths by 2050, which is more than that of cancer. So it is a burning question and burning problem, and accordingly the costs, the economic losses also increase, and it is estimated it's in pounds because it's the paper is from the England. So 66 trillion pounds are estimated to be for treating the AMR. So what is the solution? So the present problem where we have these antimicrobial resistant or multi-drug resistant bacteria, which is just eating up all the antibiotics you are giving. It's becoming superbug, and there are no newer medicines, newer antibiotics which can kill it. So what is the solution? The solution is bacteriophage therapy, which is a natural way of selectively targeting pathogenic bacteria. You can, it can kill multiple drug-resistant bacteria also. So it's like a surgical strike on the pathogens. And this is the best alternative to antibiotics. So what is the USP? USP is that they are highly specific to their hosts. They will not kill all the bacteria. They have their particular host only it will infect. They are self-replicating. They can replicate inside their host only, and self-limiting. When the host is not present, it's just like any other protein. So these properties, all these properties, the specificity, the self-replicating property, and the self-limiting property is not present in any of the other modes of treatment which are in practice, like antibiotics, herbal extracts, probiotics, prebiotics, etc. Okay, so and this is why this picture, the phage is the major a thing which can fight the multiple drug resistant bacteria. They provide a natural, non-toxic and feasible technology to control the pathogens. I hope the slides are changing. Yeah.
Yes, ma'am.
Yes, ma'am. Yes,
Yes,
absolutely.
ma'am.
Okay. So let us know now how we came to know that there are certain entities which can kill bacteria. It was in 1896. A British bacteriologist named Ernst Hankin. He was observing that in the banks of Ganges River, the people were not affected by cholera, which was rampant at that time. So only people around Ganges River were not getting this disease. And people also were like, it's like a holy water, and they were taking dips and drinking water from Ganges and all that. So he thought that there is something in Ganges water which is killing the pathogen, Vibrio cholerae. So he took the water from Ganges, filtered it through a very fine porcelain filter, and took the filtrate and added to his culture, Vibrio cholerae culture, and he saw it was killed. So he was surprised, and when he heated it and repeated the same experiment, he did not see any result. So he concluded that there is some unidentified substance in Ganges which is killing the bacteria, the disease-causing bacteria, and it is heat level. That's what his findings were, and it stopped there. Maybe after around 20 years, two scientists, Frederick Twart and Felix de Herelle. Frederick Twart was again he was a British bacteriologist, and Felix de Herelle, he was a French-Canadian microbiologist. Both of them independently discovered this entity, and it was Felix de Herelle who coined the term bacteriophage, which means bacteria eater. So At that time, this was a, because Felix de Herle, he was able to isolate these phages, purify these phages, and it was so popular. And even Stalin in Russia, he welcomed him, and another person, Eliva, he started an institute for this phage research. Okay, it was Eliva Institute for Bacteriophages, Microbiology and Virology. There he was invited and allowed to work. So a lot of phages were, produced there. And they started even treating the humans. And he suggested, Herele actually suggested that this can be given to animals and can be used in agriculture also. So at the same time, the phages, with the great, enthusiasm, people started commercializing. In 1930s itself, big companies like L'Oreal and Eli Lilly, they also commercialized these phage-based products, and with there are different products against different bacteria, for E. coli, for Staphylococcus, Enterobacteria, like that. But it was World War time, and even in the wars to prevent dysentery, these were used. At the same time, there was accidental discovery of antibiotic, penicillin, by Alexander Fleming. So this was more attractive than phage. And at that time, I think phage biology was not understood properly. The high specificity was not understood. And there were exaggerated claims of effectiveness of these phage therapy. And all these led to the decline in the phage therapy in the West. But in Georgia, where the institute was started, and in Russia, the practice started. They even produced a lot of papers, published a lot of papers, which I think was not available to the rest of the world. It was in Russian language and their language, which was not understood. But because this was interesting, the antibiotic thing was interesting, people turned to the antibiotics, and it was a broad spectrum antibiotic. Okay, all infections were cured with the antibiotics. Many bacterial diseases were cured. So the interest was lost in the West. But till today, in Georgia, Tbilisi, the capital of Georgia, the institute and the hospitals and even the counter, the drug houses have phage counters where people go and take phages for their treatment. So this is history. And now, because of this burning problem, AMR, antibiotics now have taken backseat. People have understood the misuse and abuse of antibiotics, and the interest has again increased in phage therapy. It's a new wine in an old bottle. It's a very effective therapy. So let us learn more about bacteriophages. They are most abundant biological entities on Earth. So, and they are natural enemies of bacteria, in a sense, as the name itself indicates, it can infect only bacteria, nothing else. So they are parasites of bacteria. So they will survive only inside the bacteria, their bacterial hosts, not in any other organisms. They have a very simple structure. So the genetic material DNA, which is surrounded by protein coat, and for their action, whatever they require, the syringe-like body with this neck, collar, and tail tube, they have these tail fibers and spikes with base plate. So this is a simple structure of bacteriophage. So where are they present? They are present in natural environments and artificial environments, natural environments, even including human body, animal body, wherever, and water, marine waters, soil, everywhere. And also like wastewater management plants, industries, pharmaceuticals, laboratories, everywhere. Wherever the bacteria are present, the phages are also coexist, and I can call it as omnipresent. So the phages are estimated to be in this number, that is 10 power 31 on the Earth. So these are the most abundant entities on Earth. So they are coexisting with bacteria from billions of years, and we are also living in amongst these organisms. So they are quite safe. What is the mechanism of action? So we have this bacterial host on which the phage goes and identifies its a receptor on the bacterial cell wall. Okay, that's how they are very specific. This is like a lock and key mechanism. It will not go and bind to any other bacterial surface. So its own host. Suppose if it is an E. coli bacteria, this phage will be specific to E. coli only. Okay, so this action is very, very specific, the step 1 attachment. So it particularly in particular site receptor, it will go and get attached. So after this, there is a penetration. The DNA, the genomic material of the phage is injected into the bacterial cell. So once the DNA is injected, the biosynthesis of the phage particles, the head, tail, fibers, neck, etc., are synthesized. And then these are assembled in the 4th step. And once everything is ready, the bacterial cell is lysed. It is bursted out with the phage particles. They come out and look for newer hosts. And this cycle goes on till the host is available. So in the laboratory, this is how one can visualize phages. So this is the bacterial lawn. You must have, many of you would have seen in the laboratories. So the bacterial lawn, okay, the hazy part here, and these dot-like cleared regions are plaques. We call it as plaques caused by these bacteriophages. So each plaque is derived from a single phage. So this is a method where we isolate or count the number of phages present. So this is phage titration method, and you can count the number of phages as well as you can isolate the separate. You can see different phages also in the plate. Okay. So coming to the impact of antimicrobial resistance in poultry and the products for poultry. Okay. So you are seeing the poultry farm here, a lot of birds in a shed. Okay. So bacterial infections are very common in every stage of poultry. That is, it can be, the poultry has different sectors. They have the broilers, which for the table meat. They have layers for the eggs. They have breeders for generating the old chicks. So everywhere the infection is rampant and handling so many birds, and no one wants to get the birds killed because of 1 or 2 birds getting infected. So how is the treatment done here? As a blanket treatment, the whole shed is given, or antibiotic is given to the whole shed. All the birds get antibiotics. So that is how the rampant use of antibiotics, misuse, abuse, everything is happening. In India, more than 75 people consume poultry as their non-veg food. They prefer this against the other animals, and it's a very cheap source of protein, so it's very popular. And global poultry meat, 40% of the total meat is from poultry in the world. So this is this is so widespread. So in India, every year around 8 million metric tons of poultry meat is produced. So, and the demand is growing and growing. So how to manage this demand? So all the birds are like from the day one, the antibiotics are given so that all the birds are healthy. And also there is a they have understood that the antibiotics make birds fatter also. It is called as like antibiotic growth promoter, AGPs are used. Certain antibiotics make the bird weightier. So it's used in poultry. So all these reasons make the emergence of antimicrobial resistance in poultry. And this is further spread among the birds, the handlers, and also the consumers. So today, the antibiotics are again, very, very ineffective, and newer antibiotics are not there. So this is a problem in poultry. The major infections in poultry are from Salmonella, Escherichia coli, that E. coli, Staphylococcus, and Campylobacter jejuni, which is this is prominent in other countries. In India, Clostridium perfringens is prominent. So this means some fourth or fifth position if according to the WHO list. So how are people handling this? In many countries have banned the use of antibiotics as prophylaxis or as AGPs, antibiotic growth promoters. They cannot use antibiotics. So what will they use? They will use acidifiers, probiotics, prebiotics, herbal products, etc. And after the advent of this phage-based products, even in the other countries, phage treatment is done, and FDA has approved. Initially, there was great resistance in America against these phages, but now everyone, all the countries are welcoming the phage therapy, phage treatment. So the FDA also has approved phage-based products like List Shield, Eco Shield. These are all in the other countries, Salmofresh, etc. In India, I'm proud to say that we also have products for treating bacterial infections. For E. coli, we have ColiShield. For Salmonella, we have SalShield, and E-Shield for the combined infections. Phages, which has even for Staphylococcus, this spraying on birds to bring down the Staphylococcus infections. So they're all are developed and marketed, commercialized by Aristogen Biosciences. In India, we are the first ones to manufacture and commercialize phage-based products. So what are the benefits when people use, instead of antibiotics, if they use ColiShield or SalShield, E-Shield, phages like that? The first visible thing what the farmers have observed is the reduction in mortality. Okay, and we have seen that when antibiotic fails, many a time we receive calls where they must have used antibiotics and still the mortality continues. They call and ask for the product, they use it and they are happy. So we know that when antibiotic fails, this product, the phage-based products, work. Absolutely no side effects. We have tried even 50 to 100 times of overdose. There is no side effect. And definitely there is improvement in the bird and egg quality. The hatchability is improved in the sense they keep these eggs to make day-old chicks in an incubator for 30 days till they hatch. So if there is infection, in the incubator itself, the eggs burst and it becomes waste, and the infection will spread there also. So when they have used these products, they have seen that there is improvement in the hatchability. It's quite safe and natural. And antibiotic free tag, which is a need of the customer now, is because of these products. We must have seen in the newspapers and the advertisement, people say the antibiotic free chicken, antibiotic free meat and things like that. Now you must have been seeing now, which was not there earlier. So this is how there is effect of the bacteriophage based products on against the antibiotics. So in poultry, the applications are numerous. I can say from day old chick, from egg to the meat, till it grows, meat forms the layers, broilers, everywhere. This product can be wherever there is infection, in fact. So it can be used as antibacterial agents. This is bactericidal action. Actually, Pudge has bactericidal agent. It's not static. So it can be given in. We are giving it in feed additives, and even after the bird is slaughtered. So this spraying is done in the farms as well as on the carcass. Okay, so this disinfection and food biocontrol, everywhere phage is used in poultry. So it can be for food biopreservation, food safety, decontamination, packaging, everywhere. So in the as I was saying, in Tbilisi, Georgia and all, this decontamination surface disinfection is done by phage spray. So that can be, we have started here in the farms. So to get rid of any E. coli and Salmonella infections. So this is one slide where, see, all this, what I was speaking about is the study we have done in the field studies, directly in the farms where there were 2,000 or 4,000 birds in the big farm. In the each shed we have given, and we have proven the efficacy of the product. In the laboratory, this is an experiment done in the laboratory. So we have used Sal Shield, which is against Salmonella. This is phage against Salmonella versus antibiotics. The strong antibiotics which were used in poultry, where we have used the livoquin and amoxicillin. So these discs, what we see here is livoquin and amoxicillin, and we have spotted the phage. Okay, so what we observed was, and okay, after spotting this, we have left it for days, and what we observed was the zone remained same. Zone which was made by these antibiotics remained same throughout, but the zone from the cell shield kept on increasing. The circumference kept on increasing. Okay, so this is how visibly we can see that the phages are more effective than antibiotics, number one. Number two, until the host is available, phage can keep on acting on the host. They can keep on killing the host. And why we stopped after 19th day? The plate dried up, that's all. Otherwise, we would have seen still, increase in the zone. Okay. So this is about poultry. Coming to aquaculture. Again, wherever we have mass production, mass animals, birds, etc., we have these infection problem. In aquaculture, shrimp and fish, both are a very good source of proteins, and shrimp industry is mostly it's a export oriented industry, and it generates a lot of GDP to the country. And in 22, around 9 lakh tons was produced in aquaculture. Shrimp was produced in aquaculture, And here also, Vibrio-related bacteria, in Vibrio species, various species of Vibrio bacteria are the major problem in the aquaculture. So, and also here also people use antibiotics to control vibriosis, and again there is multiple drug resistant. Now there are strict controls. I mean, there are rules. There is a authority which can take care of these use and abuses of antibiotic, or monitoring, basically monitoring in agriculture. There is some authority like CAA, coastal forgot authority. Okay, CAA it is. And so they are monitoring now. So, and fish also, which is again a popular protein giver in here also, it is 8% of global fish production in India. So 17.5 million metric tons were produced in 22-23, which is 8% of the global fish production. So here also bacterial infection, again with here it is Aeromonas, Pseudomonas species, which are problematic in fish. So which cause great economic losses, deaths and things like that. And here people use not one antibiotic, 4 to 5 antibiotics are used. And so this multiple drug resistance is a very common fact in pisciculture also. So again, as I said, the shrimp, for example, has a very poorly developed immune system. It's such a tiny, a small animal. So this doesn't have a proper immune system. So it's easily, prone to infections. But that's one problem. Because of its size, there is, repeated infections, and proper antibiotics are not there in the market. This is leading to public health issues also with antibiotic fed animals people are eating. It creates public health everywhere in any mass production. This is the problem, and majorly the export rejection because of the presence of antibiotic residues. So again, same thing: bacterial diseases, economic losses, multiple drug resistant infections. So there is a need for alternative, so which is the phage. And we do have, again, Aristogene do have products for this. And again, I would like to say, for Vibrio, the bacteriophage-based product is first in the world, I should say. I have not seen any Vibrio treating phage-based product in when I was searching. Okay, so this was the first company to develop phage-based product for aquaculture also, and these are the products. Vibrio Shield, Vibzet, and Elixir are the products which we have for vibriosis. And when people use Vibrio Shield, again, the first thing what is observed is that of the yield, better yields, increased yields. Okay, and it's very safe and natural, no side effects. Vibrios are gone, sidal effect, and okay, it's antibiotic free, so export potential is increased. So similarly, we have AeroShield for control of Aeromonas and Pseudomonas in fish. So these are the products for aquaculture. Coming to livestock. Again, I think India is the largest producer of milk. More than 300 million cattle are there in India. And the major problem faced in the cattle is the mastitis. Okay, so the causative organisms are E. coli, Staphylococcus aureus, Streptococcus species, etc. Again, it's a multiple problem. Many bacteria are causing this problem. So the losses, combining the direct and indirect effects, like economic losses, are around 30 to 40 crores annually due to bacterial infections in cattle. So for this, again, there are bacteriophage-based products. We are working, and it's in, we are testing on various forms in the cattle now. So we have tried all the ways, and the results are good. We dip, underwash, feed in water, and spraying on animals, etc. So all these are, we have observed this, it is making the animals and the handlers' life easier and healthier, I should say. So, so this is about livestock. We are in continuous research for these phages and the pathogens. Our laboratory works, our institution works continuously on this. We are collecting the pathogens from wherever possible, and we are finding the best phages, the lytic phages, the potent phages which have wide host range, and we have collected them, put them together as our product, and that's why it is successful. So I think India should not be called as AMR capital anymore. So this is our contribution towards that, I can say. So finally, let me compare the bacteriophage treatment versus the antibiotic treatment. So when we use bacteriophages, as I said, they are highly specific and targeted. Okay, so only pathogenic bacteria will be killed, or the bacteria which is targeted will be killed, and other normal flora will be unaffected, because any animal, our guts are full of bacteria only. We have a lot of bacteria which are useful, which are helping us in our day-to-day digestion or whatever well-being. So they are not harmed. When the phage is used, the beneficial bacteria are not affected at all. But again, that's why I said this is like a surgical strike. And antibiotics is not like, it's like a nuclear bomb. Just kill all the bacteria which comes in the way. So it's not at all sensitive. It is a broad spectrum. It kills all bacteria, including the required ones, the normal flora. So bacteriophage, when you apply, the intervals are flexible, because the phage goes and multiplies at the site of infection. So this, when you use antibiotic, you have to have a course. So there is a multiple application. So, and there is exponential growth in case of bacteriophage. So one bacteria killed, there are 50 to 100 phages coming out, more than that also coming out and fighting against other pathogens. Here, no such increase. So this is very important, like growing and destroying at the site of infection. So the help is available where it is needed most in case of bacteriophage. And in antibiotics, it's not like that. It's just hit and run. Just it just does its job and it is metabolized. That's it. So one phage can kill many bacteria. Here, I think one molecule should, there is a dose. So depending on the dose, it should be like, okay. And the bacteriophages are available for virtually all known bacteria. Here, antibiotics are not like that. There may be a broad, broad spectrum, but as and when you keep on using antibiotics, they become more and more resistant. and they become resistant to more antibiotics. Here there are no side effects known, and for antibiotics you see that there are a lot of side effects, including serious reactions also. And there is multiple drug resistance, which is very common when people use antibiotics and when people use bacteriophages. Continuously, if somebody uses bacteriophage, and even if bacteria becomes resistant to one phage, we have several phages which can infect the same bacteria using different receptors. So this is the beauty of using bacteriophage. And the trends is like this again. The antibiotics versus phage therapy. The efficacy reduces over the time due to resistance, but here it is sustained because the phage is very specific to its own bacteria, its host. And the environmental impact, we can find a lot of residues in all the food chain, all the food animals. Here, phage therapy, the residue as such is not there. It's eco-friendly, natural. The cost is increasing due to resistance, and when people use antibiotics, they keep on using antibiotics. This antibiotic doesn't work, use another antibiotic. Phage therapy is cost effective, and now people are adopting this phage therapy more and more. It is increasing globally, and antibiotic use is reduced due to regulations because of this AMR problem. So this is trends, antibiotic resistant trend. Okay, so what are the future prospects of phage therapy? Again, like wherever there is bacterial infection, okay, and wherever antibiotic is needed, there bacteriophages have a considerable potential for treating any and every bacterial infection. And as I said, we are with the phages from so many years, hundreds, I think it's 100-year-old therapy people have used, and they have stopped because of not understanding it properly. But because we are there, the phages are there with us, they have very good safety features. They are quite safe. And when we have used on our product, also we have seen this, there are no side effects, quite because it's natural, it's in our guts, it's in our hands, everywhere. So, and also the escalating dosages are not required for when the phages are administered. And there is a lot of understanding and advantages in the phage therapy. The phage technology is like,
टाकतात तर मी काल एका मुलीचं टाकलं होतं sample.
hello.
I ask all the participants to kindly mute your mic, and Parth sir, आप थोड़ा बाकी सारे participants को please mute कर दीजिए. Okay. Thank you. Ma'am, you can continue now. Sorry for the interruption.
Okay. Yeah, what I was telling was this phage therapy. If it is not in mainstream still in many countries, so that is required. There are a lot of advantages, and when people are like there are the phage products. That means the proteins derived from the phages, like endolysins, are very useful in therapy again. So if you don't want to use phage as a whole thing, use the protein of the phage, which can lyse the bacteria. So the endolysin of the phage, the lysine, cholins, etc., which are very useful in treating bacterial ailments. That is also in the the next step of phage therapy. The phage products derived from the phages are also equally useful and important. And even in our company, we are doing it. The cloning of endolysis also is happening in our company. And also, this phage therapy, when used, this can be used along with existing therapies. Like if we talk about poultry or poultry, animals, aquaculture, people have used along with the other therapies, like with the antibiotics also people have tried. This can be used. It doesn't have adverse effect. It can be used as complementary approach. Okay, so this has made, and the other observation is phage therapy has made the bacteria vulnerable to antibiotic. That also papers are available. So it's again win-win situation. So there are definitely a lot of prospects for the phage therapy. So this continued exploration and clinical validation is very crucial, and then only the phage therapy can come to mainstream, like what is happening in Georgia. If it has to happen everywhere, there has to be a lot of awareness about this phage therapy, the usefulness and the safety. Then I think it will be in the mainstream in modern medicine. I think this is what I wanted to tell you all. Thank you for listening to me. And I had a few MCQ as asked. So, hello?
Ma'am, we have a couple of questions.
Yes.
For this particular presentation. First of all, thank you so much for these deep insights in this particular area of the work. Like a brief overview, if I can say, for your topic to all my presenters who have joined, and just a summary, like how you have started the bacteriophage therapy, initially like bacteriophage, and then going to the advanced manner, the therapy and its importance, the comparative aspects of the bacteriophage and the antibiotics, and how the agricultural field and the animal husbandry part can also be very much covered by therapeutic aspect with the bacteriophages. It is very interesting in that way. Thank you, ma'am, for the deep insights. Now, the couple of questions which are already just popping up in the chat box, and it is very interesting to note that participants are really taking it in a deeply manner, the topic. If you agree, then we can, if you permit, then we can take the couple of questions for you.
Definitely. I
Ma'am.
think just I saw, and whether it can be used in humans. Somebody has, I couldn't, I think I'm not able to see the name, and I don't know how to open the chat box here. Yeah.
Yeah, I just,
Yes.
we technical team just tried to help you, ma'am. The
Yeah.
one question which is just, I can see, ma'am, that Dr. Abhijit Joshi is asking that the efficiency of bacteriophage therapy. He is asking about the efficiency of bacteriophage therapy, how phage can be differentiate between normal flora and the pathogenic MDR. That was his question. I repeat the question, how phage can differentiate between normal flora and pathogenic MDR.
Okay. See, the pathogens, as I said, even if I take E. coli, there are a lot of species of E. coli, and the phages very specifically find their receptors on the cell, on the E. coli cell. So the receptors are different on different E. coli, or if it is other bacteria is totally, whether it is Salmonella or Staphylococcus, receptors are totally different. So because of this very specific binding, they can bind only onto their own hosts, not on the other hosts. So that's, the receptors are the factors here.
Ma'am, the next, I hope Dr. Abhijit Joshi has got his answer. Thank you so much, sir, for the question, and thank you, ma'am, for the reply. Dr. Rupak Kumar is asking, is bacteriophage generic in nature? Is bacteriophage-based treatment or formulation regulated in India? Is any limitation is there for this therapy?
Actually, this bacteriophage therapy is not very popular. And I can say the people who are affected by this problem, like bacterial infection, which is not treated at all. If we see the literature, or if you see YouTube or whatever platform, people have told their stories. So definitely this Eliva Institute, what I was talking about in the Georgia, Tbilisi. So they, even an Indian, I think his name is Pranav. He had a severe bacterial infection, and which was not cured by any antibiotics, with any long course of antibiotics. So he went there, got it treated, completely cured of his bacterial infections. He has come back to India and he started a company. I think it is Vitalis Phage Therapy. This is the company he has started. So he will help for those who are ailing with bacterial infection for a long time, which is not cured. I think he is in collaboration with the Eliva Institute. So he's going to procure those phage. I mean, first thing, it's a a very nice process where the specific phages, because there will be a phage library even in our laboratory for any infection. It's not one phage which is used. It's a cocktail of phages which are specific for a species. Okay, so we check whether it is killing the particular bacteria. Same thing in humans. In very meticulously it is done for humans. There are a lot of regulations when it comes to human therapy. Animals, it is not so meticulous, and it's I should say it's taken for granted or something. In animal, nobody is worrying whether the animals are fed antibiotics, whether there is multiple drug resistant bacteria. Nobody is worrying. But for humans, there are a lot of regulations. So whenever a product comes for the human use, it goes through a lot of steps. So even for phage therapy, there is the pathogen is isolated, the phage is tested on the pathogen, and then only it is orally or whatever way it is given to the humans. But for external uses, I think in many places they are used, like for wounds, for spraying and all, phages are in use, even in the hospitals.
Thank you, ma'am, for the detailed, elaborative answer. I hope the speaker has got the answer. Now, the next question is, Dr. A. Madhavrani is asking that can bacteriophage therapy be used for human disease caused by bacteria? I guess you have already, yeah.
Yes, it can be used.
Specific case study, ma'am, do you have in your mind, like where you have encountered any special difference with respect to the animals treatment of bacteriophage and human treatment of bacteriophage, like if any case or any observation?
Definitely, we do have, I mean, a lot of field trials. It's not initially when we started the phage based products, when we started this therapy, we did face a lot of problems, and in a sense, a lot of questions. There was acceptance was not that good. So we had to really educate people. We had to go to farmers and explain them. They tried on small, number of animals or birds. They were happy, and then only they started using it. It's not that easy to really come to this level.
From my point of view, I am not so much of I mean acquainted with this topic, area. So that's why I'm asking, like, any major challenge you faced during the process when trial is going on, like, which is very very observable, and like it can be with the immunity and as well as with the pre-medical conditions can also be have some effect. I might maybe asking the right question, like in that way.
Yeah, definitely, because it's a flock and it's in open. Like if I talk about the poultry, it's in open. It's not like in a controlled shed or something like that. It is not like that. Maybe in some places it is there, but if you walk in, the birds will be in an open shed, and people will be walking in and out. So there are every chances of bacterial infection. There may be some preconditions also. So sometimes prophylaxis is not very effective, but treatment is always effective. So, and when they have, in the controlled environment, if the shed is well maintained and, people have the hand dip, foot dips and all, enter the shed in a proper way, the sanitation is done very meticulously. In these cases, what we have seen is when this is observed like a prophylaxis, so there is definitely the mortality reduction. See, when anybody takes antibiotics, it's just shock actually. For us itself, we take around 650 mg, but for birds,
Yes.
as a flock, they give some amount and they do get a shock. So there will be some problem in them, and their growth is affected so when compared, we have done this control studies with and without our product, with antibiotic, shed with antibiotic and shed with our product like Colishield, and we have seen difference. The mortality was very less, and compared to the birds treated with antibiotics, this we have definitely observed.
I
Thank you,
just,
ma'am.
yeah,
Yeah. Yes, sir. Please
yes,
carry on, sir. Please.
I just wanted to add a couple of points with the permission of the, respected Vasudha madam. Okay, see, I think the bacteriophage therapy is still a quite evolving field, and for clinical practice, it's not so much, prevalent or used in for human therapy, especially if you see the Indian or the Asian context. But in West, and especially in the Europe thing, it is really coming up, as ma'am has rightly said, in Georgia, and very recently now Portugal is one of the first countries in the world, okay, who have actually authenticated phage therapy as an completely verified treatment for these antibiotic resistant infections in humans. And there are certain very nice case reports, studies where especially in Australia and all, where very these kind of very critical infections, okay, where even actually an Indian, the case study is about an Indian girl there who has got a severe accident, and she was very critically injured, and then she developed later these infections caused by antibiotic resistant bacteria, which made it almost impossible to treat, and her condition worsened tremendously. Then with the application of the phage therapy, or treating with that, she has completely recovered. There are actually certain other case reports which very clearly points out to this, the effective use of this phage therapy. But as such, as if you see in the whole global context, for humans, it is still not as a, or it's been used as a verified therapy. Okay, and even less so in India. But we hope that slowly, this will also be verified or authenticated as a treatment procedure for treating infections.
Yeah, correct. In India also, as I said, there is Vitalis phage therapy company, which is, helping in human treatment with the phage therapy.
Actually,
Thank you,
ma'am,
sir. Sir, with your permission, can I take 2 more questions from the
yes,
participants?
please.
Thank you, sir. Thank you, ma'am, for the reply. Dr. Isha is asking, ma'am, any proficient and accurate method to isolate phage?
Yeah, it's a lot of, you have to really fish out or hunt for the phages. They are everywhere, but it takes time to really hunt the phages for your pathogen, whatever you want to infect, or if you want to kill certain bacteria, keeping that host, you have to really look for various samples, and with continuous research only, you can isolate a phage for your bacteria. Some, like if it is E. coli or something, maybe it's commonly present, and yeah, with it is not very straightforward, I should say.
Thank you, ma'am. Ma'am, actually, Dr. Devashish Banerjee and Dr. Abhijit Joshi and other few faculty also in our Atma University are engaged deeply in this particular research. So thank you again for taking this particular topic in this platform. I am sure that in this platform, the series of participants, they are also having in their own universities and institutions like this type of work. And I guess these particular deep insights definitely make some brainstorm thoughts to give them to in the future to take the such type of topics or such type of interesting avenues of researches to continue further. So let's not waste time and take again some questions.
Yes, yes.
Ashra, one of the participant, is asking, the therapy is very efficient, ma'am. I agree. Its specificity is great. I agree. But I have read papers which states that bacteria gets evolved upon certain infection cycle by the phages. How to mitigate that?
Yes, this is correct. As I said, as they have evolved and learned how to become superbugs, they will learn how to evade phages also. They have mechanism, but the when it comes to phage therapy, it's not one phage. That's why it is not one phage which is given for therapy. It is for the same bacteria, different phages are given. So that different phages will have different receptors on the bacteria. So if one, I already mentioned in my talk actually, even if one bacteria becomes resistant to the phage with continuous, it has to be continuously used in first case. The same phage has to be continuously used. So even if it acquires resistance, there are other phages which take care of that. So whenever it's a phage therapy, it's not one phage which is given, it's a cocktail of phages for the pathogenic bacteria. So if one phage makes the bacteria resistant, there is other phage which will kill. So it's that the phage therapy is about that, like very effective phages on a particular pathogen. It's not a single phage.
I hope madam has got her answer. Her next question with this continuation is, genetic modification of phage will work?
Yeah, the people have done some genetic modification in the phage itself to do other kinds of work, like drug delivery or something like that. It will, but they are all in still in experimental stages.
Ma'am, also there is a question by Rupak Kumar. Sir, any limitation or drawback of the bacteriophage therapy?
Limitation and drawback. Actually, there is awareness is only limiting, I should say, because people are not aware of this bacteriophage therapy. And in many, if we remember our books, it will say virus, which infects bacteria. So the term virus itself makes people scared. So this should go. This thought should go. They should understand the efficacy and the safety of bacteriophages. They are natural. So it's a natural thing, for which, likewise, I mean, so many natural things work. We know that without any side effects. This is one of those natural things we use, like turmeric or ginger or something like that. This is one natural therapy which very specifically infects only bacteria. So people have seen that they have tried to manipulate the bacteriophage to infect human cell, and they have observed that the phage could not enter the human cell, nor it could I mean, there is no multiplication. Leave about the multiplication. It could not infect the human cell. So that safe it is. It can infect only bacterial cell. So this
Ma'am,
understanding
Dr.
if it
Abhijit
is there.
Joshi.
Yeah.
Yes, ma'am.
This is the drawback.
Ma'am, Dr.
That's what
Abhijit
it is.
Joshi is asking one question. How bacteriophage can sustain in gut region? and how to characterize that bacteriophage?
So it's like any in our gut there are a lot of bacteria. Okay, so the phages also in the water we drink, in the food, everywhere phages are there. It's continuous. There is inflow and outgoing of the phage every moment. So only when there is infection or something like that, there will be change in the gut. Otherwise, it's like a normal flora. They are present just like any bacteria is present in the gut.
Surely, ma'am, it's very interesting, and the organism's features are also alluring very much in an interesting way. Ma'am, as we talked, like if you can give some insights on the industry, Aristogen Biosciences and its recent opportunities for the PhDs or the upcoming research students, like as we have long been associated from my PhD time, so I am very much acquainted with your that particular organization for my work. So in that aspect, I'm just asking, what are the kind of now the outsourcing activities, and what are the kind of job opportunities for the PhDs or the research areas? If you can please highlight, ma'am.
So at Aristogen, right now, because we are so focused and oriented towards this phage therapy, and we want to take it to the global level. That's our intention. And in this process, we have been identified or recognized for our work. There is awards given from by the government of India, and also for the aquaculture work, we do have Best Innovator Award, even from the aquaculture world. So because we are so focused and working any PhD work or the what I should say, the others faculty or any student working in our lab becomes little disruptive to us. So in future, maybe there will be opportunities where if one person can work completely on one particular bacteria, collect all the pathogens and work on phages. In future, maybe it is possible. At present, we are in the process of improving the existing products, and so we, from this post-COVID thing, we are little focused, and there are no students or such things are not happening in our company.
Okay, ma'am. Like, ma'am, like if I am not wrong, I'm just asking, like, what kind of outsourcing opportunities or bacteriophage related researches we can, or what is the way to send the samples or the type of the sample, or if you are entertaining such, analysis or these things.
Yeah, for human infections, we are not working at all. What we are working on are these three, like, for plants, yes, because of these regulations and things like that, we are not into humans at all. Okay, so wherever bacterial infections are there, except the humans, like plants, even in the agriculture, there is problems. There are bacterial infections in plants, like Ralstonia is there, Xanthomonas is there, which is causing huge economic losses because of the infection. So there we are planning to work. And what was the question?
Ma'am, the sample, the type of the sample, if you wanted to send, or the way the sample we should send you, in which form, like if any analysis we wanted to do in that way.
Yeah, the sample analysis is like, if it is just like bacterial identification and characterization, such things we can do. The samples has to be like from if you find any pathogen of these animals, whatever fields we mentioned, if you find a new one, it can be sent as a plate with single colonies, or the tissue can be sent. We can isolate the pathogen, or if you have a pathogen by itself, you can send a stab. So, and there is one more question I'm seeing from Ishita.
Yes, Ishita ma'am is asking, are there any changes of any immune response generation in human or animal body due to bacteriophage therapy?
This bacteriophage therapy, like in animals, what we have seen is like in humans, yes, their papers say that there is some immune response. The thing is, when you administer phage, it is like enters the system, and if the bacteria is there, it hooks on there, and otherwise it is coming out of the system. So the time for the generation of immune response is too less. Maybe continuous use may generate something, but otherwise it is not so. The system may reject, but uh, it is very. I have seen in 1 or 2 papers about these. But when we have used in our animals, what we have seen that when we feed the animals with the phage, like phage treatment is going on, we have given some phages. When there is infection, we have seen that in the feces of that particular shed, we have seen more counts of plaque forming units were more. That is, the phage has increased because of the host present there. If the host is not present, is directly out of the body. Same numbers of phage. But when the host is there, there is increased number of phage in the feces of the bird, of the animal.
Thank you, ma'am. Thank you so much. I hope there is no more questions, and if there is, we will share in this particular link the mail ID of Dr. Vasudha ma'am. Truly speaking, the topic is still holding so much of questions which we can address later, and it's a never ending topic, questions which are coming, and we can see the chat box. I hope my coordinator sir will agree with me, but still we have to start the next session. Our next speaker is ready. So with this good note, I would like to congratulate again Dr. Vasudha ma'am, and we are deeply privileged by your presence and these deep insights. पार्थ भाई, थोड़ा सा ताली हो जाए इस बात पे. We can just acknowledge here her commendable research experience and the vast area which she has covered. Parth bhai, little bit.
Thank you so
Ma'am,
much.
Thank you, ma'am. So now you
Thank you,
can
ma'am.
be there, or you can also just take the session lead, ma'am. It's up to you. Dr. Devashish sir, can I hand over you the next session?
Yes, ma'am. Yes, please.
Thank
you. Okay.
Thank you, ma'am.
Thank you so much, Moshi madam. And so it is my utmost privilege again to welcome our next speaker, okay, for this session. Before that, I just wanted to request all of you, all the dear participants who are joined in this program, for a group photo once more. So, if it is not very bothering, I request all of you, if possible, to kindly switch on your video once more so that we can have a nice group photo. This is actually good so that we can see each other in a common platform, okay, and have it as a memory with us, for quite some time. Also, it is important for, some kind of documents regarding the FDP program to submit to the AICTE. Okay, so once again, all of you who can kindly switch on your video, please do that so that we can have very nice, happy, short group photo.
Yes, sir. Participants are opening their video, and
Yes.
it's very nice to see in the chilling night of
Thank
winter,
you, Rupak sir.
January month.
Vipul sir.
People are still. Thank you so much,
Vijay sir.
participants. Your presence is very
So good
valuable
to see
to
all
us.
of you. I request the other participants also to please get some motivation by seeing your producer, and please switch on your video.
Participants, we can have a nice shot of this moment. So we request you kindly.
It's actually good to see each other also, so that you can see once for a while, because this is the only platform we are all joining, but our videos are off, so we don't see each other. So,
We also request the resource persons, if they are there in the forum, please on your camera, so that your presence with our respected participants also can make this moment like a lifetime remembering moment.
yeah,
So
Parth
I
sir,
also request the resource persons, please.
पार्थ सर आप थोड़ा वो लेते रहिए.
Yes, sir. Yes, sir.
Great view लेते रहिए पार्थ भाई.
Thank you.
The pictures.
And we are really grateful to all the participants for switching on their videos.
Rushi sir, if you can please on your camera.
No, I think there is some kind of technical problem for Rushi sir. Because of that, he might not be able to actually do that.
Okay,
So,
okay,
yeah,
no issue, no
so
issue.
it's okay. So whoever can,
Participants,
it's
still
fine.
some photos are not coming. We are eagerly waiting for your glimpse. Please put on your camera. It's a humble request. Maybe we don't get this type of moment in the next tenure of time. So it is a great privilege to have you all in a single platform.
I think that's fine. Maybe
Yeah.
these all are not in a comfortable
Yes.
position to
Yes. पार्थ भाई आपने शायद ले
video.
लिया photos. I hope so.
Yes,
So-
thank you, ma'am.
Thank you so much, participants. Thank you so much, ma'am. Thank you so
Thank you
much.
all. Thank you all.
Thank
Now,
you, coordinator sir.
yes,
So we can go to the next.
you can put off your video now. Yeah, if you like. Yeah, thank you. So, it's my great privilege again to announce the next speaker in this third day of the 6-day online Atal FDP program. So the next speaker is Dr. Rushi J. Bhatt, another very eminent personality, scientist with us. And his topic, that what he's going to deliberate upon today, is LC-based untargeted metabolomics, a power tool for identifying potential biomarkers for early disease detection. Before he actually starts his talk, I just wanted to give a brief overview of the credentials of Dr. Rushi J. Bhatt sir. He has more than 18 years of experience in biotech and pharma industry, which is really incredible. And he has worked on many industries till now. Okay, I think totally maybe 9 to 10 industries or even more till now. At present, he's engaged as a team leader and senior scientist at Inaugural Biotech. He has a profound experience on more than 25 fermentation technologies, types, semi-synthesis and peptide-based products like antibiotics, vitamins, etc. He has an excellent background in product development, process, and scale up to the manufacturing stage. He is managing many kinds of units and departments in industries, or has been managing that, like microbial fermentation teams and labs and so on. He has a great knowledge about various guidelines like CGMP, US FDA, WHO, and many others. He has a great professional and achievements in pharma industry, like he has got many kinds of awards, accolades, achievements. He has a profound knowledge in many high-end instruments like HPMV steam sterilizer, antibiotic zone reader, microscopes, and so on. So with this brief background of Rushi sir, I invite you to kindly deliver your eminent lecture. मैं पार्थ भाई को ऑल्सो रिक्वेस्ट करूंगा कि आप रूसी सर को काइंडली प्रेजेंटर राइट्स दे दीजिए. Rusi sir, I think you have the presenter rights now, so you can present your slides now. Thank you.
Good evening, all of you. Good evening, all of you. My name is Dr. Rishi Bhatt.
Good evening,
I
sir.
did my PhD in biochemistry. Hello.
Yes, sir. You're audible, sir.
Okay. I did PhD in biochemistry and I have total 20 years experience in fermentation and fermentation-based API. And today's my topic is LC-based untargeted metabolomics, a powerful tool for identifying potential biomarkers for early disease detection. What is metabolomics? Is a rapidly growing field. Metabolomics is a rapidly growing field that aim to understand the complex metabolic pathway involved in health and disease. Liquid chromatography mass spectrophotometry is an analytical method based untargeted metabolic has emerging a powerful tool right now for
Just
investigating.
one thing. Sorry to interrupt you. Are you presenting the slide, sir, or you are just telling that orally?
Hello?
Sir, are you presenting any slides, sir, or you are just telling that orally?
No, I'm presenting slides.
Okay, but sir,
I have shared to you.
No, but it is not actually visible, sir, the slides. I don't know others can see that or not. Moshe ma'am, can you just confirm whether the slides are visible or not?
No, sir. No, sir. It is not. It is not.
Okay. Let me.
Rushi sir, please give the sharing option. If you are using laptop, at the bottom, there will be the television sign. That is a sharing option. And if it is a mobile, that is also one television. On that, one arrow is there, a top arrow.
Okay.
Just click it.
Yes, sir. Yes,
Yeah.
now it's working.
Yes. Now share the PPT, sir. Thank you, participants, for your spontaneous response. We are very much inspired by your continuous presence and the continuous touch. You are there with us. You are the strength of us. Thank you so much.
So my topic is LC based. Now it is visible.
No, sir. You just open it, sir.
So you
And
just
then
have to
share
open
I
it.
have already opened it. Share it.
Sir, do one thing. You share the window tab. Maybe it is a network, sir. Please come to the smooth network.
मैडम यहाँ पर बता रहा है मैंने शेयर किया वो.
Sir, okay. Devashi sir, can you see?
No, not yet, ma'am. Sir, your screen is visible. Okay, your desktop screen is visible. Okay, we can see that the slide is there, but there are some other things also. You have to click and just open that. सर अगर आपने ऑलरेडी किया है मे बी आप एक बार उसको एक स्टॉप शेयरिंग करके फिर से एक बार करके देखिए.
थोड़ा नेटवर्क इश्यू है यहाँ पर. मैं कंपनी में हूँ सर, इसलिए.
सर हम पार्थ भाई को बोल देते हैं, वो शेयर कर देंगे.
ओके, उनके पास है मैडम
जी उनके पास है जी. ओके सर, आप स्टॉप शेयरिंग कर दीजिए ऋषि सर. हम अपने टेक्निकल टीम को बोल देंगे, वो कर देंगे. ठीक है सर.
ओके.
नॉट एन इश्यू. पार्थ भाई, आप ऋषि सर के पावर पॉइंट को प्रेजेंट कर देंगे.
यस मैम, वेट अ मिनट.
जी थैंक यू सर. थैंक यू. ऋषि सर शेयर हो जाए, आप नेक्स्ट बोलिएगा तो पार्थ भाई नेक्स्ट करते जाएंगे.
Okay.
It has come now, sir. You can start now.
Yes, it is visible. Thank you, Parth bhai.
Thank you.
Sorry for interruption. Good evening, all of you. My name is Dr. Rishi Bhatt. I did my PhD in biochemistry. I have total 20-year experience in fermentation and fermentation-based API. And today's my topic is LC-based untargeted metabolomics, a powerful tool for identifying potential biomarkers for early disease detection. Next slide. Parthbhai, next slide. So metabolomics is a rapidly growing field that aims to understand the complex metabolic pathway involved in health and disease. LCMS is a powerful technique based on untargeted metabolomics as an emerging powerful tool for investigating the metabolic changes associated with various disease, for identifying potential biomarker for early disease detection and treatment and monitoring. Next slide. Parthbhai, next slide. The latest advancement in LC technology and data analysis technique have facilitated the more accurate and sensitive detection of metabolic in biological sample. The clinical application of untargeted metabolomics are emphasis include its utilization in disease diagnosis and treatment monitoring and personalized medicine. Examples are provided how biomarker of various diseases such as cancer, CHD means coronary heart disease, and infectious disease have been identified through the application of untargeted metabolomics. Next one. The latest advancement next slide, please.
पार्थ भाई आप चेंज कीजिए स्लाइड को. Yeah. Sir, I think slides change हुए हैं अभी. You can continue now.
वहां पर अटके हुए हैं सर. No problem.
Yeah.
Now let's discuss about LCMS. LCMS, what is LCMS? LCMS is an analytical chemistry technique that combines physical separation, capability of liquid chromatography, mostly say HPLC, coupled with the mass analysis capability of mass spectrophotometry. LCMS is a method of physical separation in which components of liquid mixture are distributed between two invisible phases. One say stationary and another one is the mobile. And mass spectrophotometry is an analytical technique that measures the mass to charge ratio of the charged particle. Although there are many different kinds of mass spectrophotometer are available, all of them use electric and magnetic field to manipulate the motion of the ions produced from an analyte of interest and determine their M/Z ratio. What are the advantage of LCMS and disadvantage of it? Advantage is highly sensitive and specificity, ability to detect and quantify a wide range of metabolites, and disadvantage complex sample preparation and high cost of instrumentation and maintenance are needed in this one. Before starting LC analysis, before 24 hours, we have to maintain temperature of the room. Now, in metabolomics, there are two types of metabolites, targeted and untargeted. What is targeted? Analyze a definite set of metabolites that are chemically characterized and biochemically annotated. This approach is effective for testing specific hypotheses. What is untargeted? Analyze all measurable analytes in sample, include chemical unknowns. This approach is often used in hypothesis generating studies such as biomarker discovery. Focusing only untargeted metabolomics. Untargeted metabolomics is a method are global in scope and aim to simultaneously measure as many as metabolites as possible from a biological sample. Biological samples, suppose किसी को किडनी रिलेटेड प्रॉब्लम होने वाला है डायबिटिक के कारण, तो हमें चेक करना है, तो उसके यूरिन सैंपल हम चेक करेंगे, बराबर। तो बायोमार्कर वो बताएगा हमें तो कब होने वाला है, तो उससे पहले हम क्या कर पाएंगे, उसका डिटेक्शन, उसके रिलेटेड ड्रग्स हम उसको दे पाएंगे. दिस अप्रोच अलाउ आइडेंटिफाइंग मेनी मेटाबोलाइट previously biochemical defined or not. In contrast to targeted metabolite analyte, it is not possible to achieve precise quantitation of these metabolites. One of the disadvantages of untargeted analysis is that hundreds of metabolites can be detected simultaneously. Compound identification relies on use different library. Here, different type of library is available for detection. Now, what type of sample preparation is for this untargeted metabolites? Suppose serum and plasma. First of all, we have to throw the sample in ice, add deuterium labeled internal standard, add methanol in 1:3 ratio, precipitate the protein in serum and plasma, centrifuge, centrifuge, then filter and lyophilize. Now, this lyophilized one reconstitute when LCMS analysis is going on. Now, if sample is urine, so how to extract biomarker from urine sample? Take 50 to 100 microliter and in chill condition, vortex in methanol, SCN mixer, centrifuge, filter, and lyophilize, and analyzed to be analyzed when and reconstituted when used in LCMS. Now, what is the clinical application of this untargeted metabolite? The clinical application of untargeted metabolite has a potential to revolutionize the diagnosis and treatment of many diseases, including cancer, metabolic disorder, cardiovascular disease. Some specific example of clinical application of untargeted metabolite include cancer, infectious disease, diabetes, cardiovascular disease. Overall, the clinical application of untargeted metabolite hold the great promise for improving diagnosis, personalized medicine, drug development, and lifestyle intervention. In cancer and untargeted metabolites, one of the hallmark of cancer is metabolic reprogramming, which refer to change in how cancer cells use the energy and nutrient to support their growth and survival. Metabolic changes in cancer can include increased glucose uptake, alteration in glucose metabolism, change in amino acid metabolism, fatty acid uptake and synthesis, and change in mitochondrial function. Understanding the metabolic change in the cancer cell is essential for developing a new diagnostic and therapeutic strategy that target these processes. Early diagnostic of tumor classification and response to therapy and tumor metabolism were some specific example of clinical application of untargeted metabolomics. The metabolomics profile of cancer is often distinct from normal cell, making it possible to identify metabolic biomarker that can be used in early cancer detection. Untargeted metabolite can be used in coronary heart disease also. Several studies have shown that patients with the CHD have altered their lipid, glucose, and amino acid metabolism. An alteration in the metabolism of the D-arginine and ornithine glycolysis, oxidation, degradation of branch chain fatty acid and sphingolipid metabolism have been linked to coronary heart disease. It has been discovered that serum oxynurin and triglyceride are candidate of biomarker for the early diagnostic of coronary heart disease.
Excuse me, sir. Just 1 second. I think one of slides are moving a little fast. Okay, so I think you've already finished your talk, right?
Yes, yes.
Okay, fine, no problems. Yes, sir. Yeah.
Okay.
Please continue, sir.
Future of this untargeted metabolism is promising and rapidly growing. In research, By identifying changes in metabolic profile before the clinical symptoms appear, we can detect disease and their early stages when they are most treatable. This could lead to more effective treatment, improve patient outcome, and reduce the health cost. Thank you.
Thank you, sir. We are really grateful to you, sir, for this wonderful talk on the future prospects of metabolomics. So in your very nice talk, you have touched upon very important points like the how important is the metabolic profile of individuals, especially in certain diseases. Then you have talked about LC mass spectrometry, the how the different targeted and untargeted analytes are important in the characterization, the diagnosis or the treatment of any disease. Then you have shown how especially the untargeted metabolomics can be analyzed and used for the treatments of many kinds of diseases like cancers, coronary heart disease, and so on. Okay, so we are again very grateful to you for taking your time out and delivering this nice lecture. Parth bhai, आप एक वो let's all of us have a good round of applause for sir. So Parth bhai, आप kindly वो ऑडियो क्लिप थोड़ा प्ले कर दीजिए सर के लिए.
Thank you. Thank you.
Now, sir, actually the floor is open now for discussion and few question answer round. So there are, sirs, already some questions are coming in the chat. Okay, which really shows that your short talk has also ignited the minds of the participants. So if you allow, sir, can I read out to you some of the questions?
Yes, sir.
Okay, so sir, Abhijit Joshi sir is asking how to differentiate polar and non-polar metabolites.
Pardon, sir.
Sir, Dr. Abhijit Joshi is asking how to differentiate polar and non-polar metabolites. सर, आपको समझ में आया सर क्वेश्चन? मैं और एक बार बोलूं सर? Okay, maybe we can move on to the next one. I think there is some kind of internet connectivity issue with Rusi sir. So Rusi sir, the second question comes from Ajit Kumar sir. He's asking, is there any possibility to detect detect using GCMS? Means yes. If yes, it means which one? Okay, I think the question is little ambiguous. I think he's trying to ask that is there is any possibility to detect the same things like maybe the untargeted metabolite through GCMS, and if it is so, which is best, LCMS or GCMS?
Yes, sir. Certain metabolite we can do in GCMS also, but amongst this, both LC is more powerful because large
Okay.
number of metabolites separate by LC only.
Okay.
Only
Okay.
volatile metabolite we can do GCMS.
Okay. Thank you, sir. Sir, the next question is again from Dr. Abhijit Joshi, sir. He is asking, can we detect new metabolites from LCMS as it is purely library based? That's why he's asking this, that whether new metabolites can be detected from LCMS or not.
New metabolite can be detected in LCMS.
Okay, so can you throw some light on more on this question? Like he's asking that because it is purely library based, that means I think he's meaning that you have to take the reference of some kind of known compounds. Okay, and how you can actually identify the new metabolites, like any new or which is not reported yet.
Not reported, then we have to do some by prediction based only we can do it.
Okay, okay. Thank you, sir. Sir, and then the next question is from Dr. Lakshmi Yadav. She's asking that can we detect tumors from metabolic? Okay, again, I think the question is not complete, but what she's trying to mean is that can tumors be detected by these kind of characterization?
Yes, tumor can be detected by this kind of characterization because tumors pathway are altered. So by diagnosing these body fluids, serum and plasma, we can diagnostic that tumor in early stage.
Okay. So next question is from Dr. A. Madhavarani. She's asking that what type of samples are needed for the early diagnosis of cancer? Is it tissue biopsy? That means if tissues are needed, or any other kind of samples are enough to detect the early stage of cancer?
First of all, body fluids. By blood sample we can detect if it is metastasized. Otherwise, localized tissue we needed.
Right. Sir, one last question is there, again by Dr. Lakshmi Yadav madam. She's asking that how can she detect the severity of cancer through this kind of analysis? Meaning, is there a way to detect the severity or the advanced stage of the cancer through this analysis?
Yes, we can check early stage of advanced stage of cancer.
Okay, fine. So I think that's all the questions that we have, and I thank you, sir, for very nicely, giving the responses and the information about all the questions that were asked. I think the participants are satisfied with it. So once again, sir, we are really grateful to you to taking time out of your busy schedule. And I just like to inform to all the participants that Sher is still, there today on Sunday. He is so busy that he is still in the company working now.
Right now in company. Right now I'm in company.
Yes. So, and out of that schedule also, he has taken some time out because he has committed to that. So that shows his the level of his commitment also to deliver some important points for you. So I think on behalf of all the participants, the coordinators, and the entire FDP team, and also the university, we are really grateful to you, sir. Thank you for taking the time out.
Thank you, sir.
Thank you, sir. So
can we have a round of applause? ताली हो जाए थोड़ा सा. पार्थ भाई.
हाँ मैम. Wait.
थैंक यू सर फॉर योर कमेंडेबल परफॉर्मेंस एंड
Thank you,
आप
madam.
इतने
Thank you,
बिजी
ma'am.
शेड्यूल के बावजूद भी हमारे साथ जुड़ पाए. सर किस तरह से आपका शुक्रिया अदा करूं समझ में नहीं आ रहा है. थैंक यू सो मच सर. थैंक यू सो मच.
Thank you, madam. Thank you.
Okay, so with this, we have come to an end of this session. I again invite my respected coordinator, Dr. Moshui ma'am, to conclude the session for today. Thank you,
So
Parsa.
as we are at the end of the session, now it's time to wind up the Sunday night. So we have brilliant two speakers with us today. The first one was the bacteriophage therapy. Its advancements and its real-time exposure applications in poultry and as well as aquaculture. And that was the tremendous sharing which we have got. Maybe our participants also agree that you have got very good insights. The next speaker was our again one marvelous presenter, that is Dr. Rushi Bhatt from Endoc Life Biotech in Rajkot. So we have a good exposure of new technology in the pilot scale fermentation process, how it works. And being a bioprocess practitioner from my side, I'm very much delighted to personally feel the presence of speaker in this particular session. So I hope participants will agree. They have got some new avenues of researches to think about for their students and for themselves. So keeping these things, I once again make you remind that feedback and attendance has been shared in this particular Cisco WebEx link chat box. Attendance will be closed today only, but the feedback will be remain open tomorrow till 12 noon. And your feedback is very important for us. Please don't miss that. Please give us the feedback so that we can improvise ourselves. The two speakers, we are heartily acknowledge your presence. We are really indebted for your presence. Thank you so much. So with this, good night, Shaba Khair and Shubhratri. We once again going to be joined tomorrow in the same time and again with the beautiful topics. Thank you, coordinator sir, for constantly keeping in touch and coordinating with me. Sorry for the inconvenience of the network facility which I'm having. Parth Bhai,
Yes.
thank you so much at the backend team support, and the CRIT director sir for their tremendous support, and the whole Atme University management for their tremendous support. Participants, you are always our strength. Please don't go from this particular forwarding meetings and forwarding links. Your presence will always motivate us and always strengthen us, our strength. Thank you so much. Good night. With this, we are going to shut down this particular link. Thank you. Have a good night and have a good Sunday till what is remaining. Thank you.
Thank you all.
Coordinator
See you
sir,
again
I'm
soon.
taking leave. Thank you,
Yes.
coordinator sir. We are
Yeah.
leaving.
Thank you, ma'am.
Thank
It
you,
is my
resource
greatest
persons.
privilege. And thank you really
Thank
all
you.
the participants from the bottom of our heart for your, active participation and joining. We hope to see you again in the same spirit for the remaining days. Thank you so much. Take care. Good night, everyone.